Okay, so good afternoon, everybody. Welcome to the course for immunology for 2024. So, as you already know, this is a 12-week long course. I am Ankita Bhattacharya. I'm a PhD student at IIT Kanpur, and I will be working as a teaching assistant on this course. So, every week we'll be having these discussion sessions. So, the basic idea behind it is to clarify any doubts that you have regarding those week's lectures, any questions that you might have apart from what has already been taught in the lectures, something additional that you want clarified. If there are any topics that you want to be discussed, please bring them up. If I have the material ready for it, we'll discuss it that day, or we can bring it up in the next class. So this is overall a discussion and doubt clearing session. But apart from that, I also have some slides prepared in advance so that we can go over, just brush over the topics that have already been discussed, in case there is some additional information that I want to share with you that is related to what already has been discussed by the professors on that course. So that is the broad outline. At any point of time, if anybody has any doubts, please either raise your hand or just unmute yourself and ask a question. There's no need to wait till the end of the session. Any topic that you think is difficult, that you need more material on, I can get more schemas, more slides, etc. So that is the basic structure of how we'll be conducting these sessions. So with that in mind, we'll get started with the course. So assuming that my slides are visible, so what you see here in the first slide, which is basically just, I think there's some issue here. Just give me a second. Yeah, okay, should be fine now. Okay, so what you see here are the different types of cells, just schematic representation of all the different types of cells that comprise the immune system. Um, Now, I'm assuming that everybody comes from different backgrounds. Your educational background might be different. But one thing to keep in mind is that because immunology is not a very preliminary course, not a very um, not for students who don't have any background. In case somebody has some difficulty who has not studied molecular or cell biology in advance, please let me know. I'll get into details about those topics because you need that background information to be able to understand how the immune system works. Keeping all that aside, we will try to keep the course discussions simple and easy for everybody to understand, but please interrupt me at any point of time if required. So, first of all, the first week, all of the lectures are giving you an overview of how the immune system works. Particularly, I would like to stress on the fact that this is called as the immune system. System being because it is not just one particular cell or one particular organ which is responsible for conducting the immunity or for maintaining the immunity of an individual, but in fact, it is a combination of multiple organs, multiple cells, cell types, and components of those cells that are all together mediating the immune counterpart that allows an individual to survive despite all of the different types of infections, pathogens, viruses, etc., that are surrounding us. So that is the stress. That is the reason why the immune system is being called as a system and not just as a singular component. Again, let me reiterate that this is a discussion session. It is not something that I need to continuously keep on talking. Please interrupt. We can have a back and forth. We can discuss any point that you want to be reiterated. So here, first of all, immunity, as you can see, is defined as the state of protection from infectious diseases, and it is derived from the term immunus, which means to accept. So it is a defense system of any individual. The question that might come to your mind is that here we are particularly looking at only the human immune system, right? But if you think about it, every organism, almost every organism, has an immune system that allows it to survive and allows it to bypass all of the external interruptions that exist in the environment. For example, let's talk about bacteria or microorganisms in general. So these organisms also have their own defense systems. They are not as complicated or as evolved as the human immune system, but it does exist and it does allow the bacteria to survive under the conditions that it is exposed to. In fact, if you have some background regarding recombinant DNA technology, then you would know that the system defense that exists in bacteria is actually utilized for recombinant DNA technology. So the restriction enzymes that you might be familiar with, they are part of the immune system of bacteria. So it is less evolved, but it does exist. And it comes from the basic need to survive against all challenges, right? So that is state of immunity. Now coming to the fact that what exactly does the immune system do? So it has two main jobs, A, to recognize and B, to respond. Why? Why is recognition important? Recognition is important because response comes after an intruder or something different from the normal system is identified. In fact, the recognition part of the immune system is extremely critical. Why? Let's talk about the fact that suppose if the body is not, if the immune system of an individual, of any particular individual, is not able to identify that there is an intruder and starts considering its own system as an intrusive entity or as an external entity, then it leads to diseases. They are most commonly identified as autoimmune diseases, which is something you might have come across in the lectures as well. So one example of that is what happens during AIDS as well, HIV/AIDS as well. So there, in such individuals, their own immune system starts attacking the body cells. As a response to that, that individual becomes very immunocompromised and very susceptible to a number of other diseases. They are not able to counter even a simple cough or cold or very, very basic diseases, which any other normal individual would be able to do. And as a result, they do not survive, and it becomes a very it is almost always lethal. Such individuals are able to survive for some period of time, but HIV/AIDS has not really been cured till date, even though there's a lot of research that is going on about it. So that is why the ability to recognize that there is something external is very critical. So as part of recognition, let me just get this. So as part of recognition, the first thing that you see here is that it needs to be highly specific. The immune system should be able to identify differences even among pathogens. Why? Because every pathogen or every intruder, whether it be a bacterial system or some sort of parasite, a virus, or any other pathogen for that matter, they all have different compositions, different identifying markers on their surface, which the immune system is able to recognize. Why is that important? Why can't there just be a generic response? In fact, there is. There is a primary response, which is very generic, which is what we'll come to, and that forms part of the immune system. But that innate immune system, is not very efficient. It helps in countering the initial problem that the body is facing, but for sustaining a response or for providing a response which is long term and which is helpful for the individual in the long term, the specificity factor becomes extremely important. Therefore, the immune system should be able to identify and recognize even very subtle differences, as has been mentioned here, that distinguishes one foreign pathogen from another, even if they differ by something as little as one amino acid. Secondly, it should be able to distinguish between foreign molecules, or whether it be some external pathogen, or it is part of the host system that is different from other cells of the host system. For example, when that individual might be infected by some virus, or when it is when it is a cancerous cell, a cell that has undergone transformation, neoplastic transformation, and has become cancerous and therefore is very dangerous for the body, then it becomes non-self and should be able to distinguish it against the normal cells and normal proteins of that individual, which is called as self-non-self discrimination. So this is one of the most characteristic features as part of the recognition component of the immune system. That is, it should be able to carry out self-non-self discrimination. On the left, you see a representation of some markers of non-self. For example, in case of bacteria, you see these are sort of cocci or round-shaped bacteria, which all have some very distinctive markers on their surface, and these are identified as non-self markers. And on the right, you see the example of SARS virus. Of course, the most recent example is of COVID. So the coronavirus spike proteins were actually used to identify, were used even as a vaccination strategy for that matter. And they were able to design that vaccine because that helped sensitize the immune system of healthy individuals to that pathogen, so that in case there is a future infection with that pathogen, with that virus, then such individuals would be able to counter it and would be able to survive the same. So that is the recognition component of the immune system. Then comes the response. So once that foreign organism has been recognized, the next job of the immune system is to recruit a response or to recruit cells that would be able to would be able to provide security against that identified pathogen. And that is called as the effector response. Why is that necessary? That helps in elimination or that helps in neutralization of the organism. When we say elimination, it means that either that organism would be killed off completely, or it might just so do that the organism is no longer able to replicate. The amount of that pathogen within the system is brought to a standstill. It is no longer allowed to increase in number, so the body can then tackle that infection at a much quicker rate. Not just this, this is the initial response, which is called as the effector response. Next comes the memory response. Let us look at the graph for understanding how that works. So this graph is representing the concentration of antibodies against time. Why antibodies? Because antibodies are part of the memory aspect of the immune system. When we say memory aspect, it means that the immune system, apart from being able to carry out the self and non-self distinction, it is also able to retain information that it has once come across. So which is actually forming the basis of vaccination as well. So a child that has been vaccinated for a certain pathogen, let's say, for example, hepatitis, when children are born in the first 6 to 8 months, a large number of vaccinations are usually carried out. That helps in sensitizing the immune system of such a small individual who has barely encountered any infections in their life, so that their immune system can start building up. And sometime in the later years, if that particular person encounters that same pathogen, they would be able to counter it much quickly and might even be able to bypass the actual disease. So this is the basic idea that comes from the memory aspect of the immune system. So later exposure to the same foreign organism can induce a memory response, and this is a component of the adaptive branch of the immunity, and that helps in generating an A, very rapid, and B, very heightened immune reaction. So if we again come back to the graph, what you see here is that when there is an initial exposure, did somebody unmute? If there's a question, please let me know. And if I'm going too fast, please let me know that as well. I'll slow it down. Okay, so when you see with respect to time, when a particular individual encounters an antigen for the first time, what do we mean by antigen? Antigen is a particular molecule that is able to elicit an immune response within an individual. Elicit as in it is able to trigger an immune response in an individual. So for example, let's talk about the chickenpox virus. So when a particular individual gets infected with the chickenpox virus, which is varicella zoster, for the first time, then there is an initial response to it, which allows that individual to recover from that disease. It takes time, around 2 to 3 weeks is the usual recovery time for chickenpox virus. And as a result to able to as a result of that exposure, there is a generation of antibodies that will allow this individual to tackle a secondary exposure to that same virus again in the future. What happens is that when there's an absence of that virus in that individual after they have recovered, say after 2 to 3 weeks, then again the level of those antibodies goes down. However, it does not completely become zero. There is a very basal level of antibodies that is maintained within that individual, so that in future, if required, they would be able to tackle the same. Now, when there's a secondary exposure to that same antigen at some point of time, then you see that the response that is picked up is extremely quick and is a very, very heightened immune response, which means that they would be able to tackle that disease much quicker. So this is the memory response of the immune system, which is much more rapid and much more heightened in nature. There's some issue in the lobby. Okay, so keeping this in mind, here you see a representation of here you see a representation or a tabulated format of all the different Nobel laureates who have received the Nobel Prize for their contributions towards the field of immunology and the field of um, vaccination, immunology, building resistance towards diseases, etc. Because it is such a huge contribution to mankind, a large number of Nobel Prizes have been awarded. So you see, research began as early as 1901, and Emil von Behring was the first person who actually started studying the serum. And from there, he identified that there was a component of the serum that is actually able to sensitize individuals to certain diseases, which later on was identified as antibody much later into the future. But he was one of the first people who was able to do that. And supporting him was Robert Koch, who was able to bring about cellular immunity to tuberculosis. Then we look at Mechnikov and Paul Ehrlich from Russia and Germany, who were together able to look at the role of phagocytosis that occurs and the role of antitoxins that are present as part of the innate immune system. Then we look at Charles Richard for anaphylaxis, Jules Border for complement-mediated bacteriolysis. We will be going through all of this as part of the course. These are very elaborate topics which would be taken up in individual lectures, and also we'll be discussing it in our sessions as well. Then the reason I'm trying to introduce this is because these are important for your basic knowledge, and people getting signed out for some reason. And also because these will quite be very useful for competitive exams if you eventually want to appear for the same, and obviously for your own knowledge as well. So Karl Landsteiner was the scientist who actually brought about the concept of human blood groups, how the blood groups differ from each other. I assume that you must have studied it at some point of time in your preliminary, either in your schooling or your initial college years, where you know that the different blood group types or blood types that we know as A positive, B positive, AB, and AB negative, and O positive. All of these are a result of the different types of antigens that are present in such individuals. And we also now understand that any individual cannot donate blood to any other individual. There has to be blood type matching, which must be performed. Otherwise, it triggers an immune reaction so severe that such people will not be able to survive. So this landmark discovery was performed by Karl Landsteiner, and you can look at the other details further as per your own convenience, like the discovery of antihistamines, which we buy so easily from pharmacy shops nowadays. For example, commercial names like levocetirizine or Allegra for that matter. These are all antihistamines which are able to tackle that initial allergic reaction and are able to calm down the immune system of people who are very susceptible to these kind of allergies. Um, the chemical structure of antibodies is something we'll come to eventually. We will briefly be discussing the major histocompatibility complex today, monoclonal and polyclonal antibodies, etc., transplantation immunology, the gene rearrangement and antibody production by Susumoto Nagawa, which was one of the most landmark discoveries, which we'll be discussing in great detail eventually in the upcoming weeks, because it is a very complicated and very beautiful concept that you must understand. And just a few more examples for your own reference. Now, moving forward from this, yeah, what we now will look at are the major two branches or the major two subtypes of immunity, which we commonly know as the innate and adaptive immunity. Now, the word innate by itself means that it is present by birth. It is not the result of any exposure or any external influence, but it is present as a result of the of an individual being born with a basic immune system. It is considered as the first line of defense. Secondly, why is that important? That is because whenever a pathogen comes in, the first bodyguards that are present is the innate immunity. It might not necessarily be sufficient to tackle that particular pathogen, but it does come into play. It prevents infections at its very outset and is able to work very quickly within a short time. So that's why it's considered as the first line of defense. It is able to eliminate pathogens within a few hours of encountering it. It is able to carry out self, non-self discrimination, but it is not capable of recognizing the very small differences that are present between foreign molecules. Now, if you remember in the first slide, we had said that the two characteristic features of the recognition aspect of any of an immune system involve self non-self discrimination and the ability to distinguish among pathogens. So while innate immunity can do the former, it is not able to, it's not capable of carrying out the distinction between the subtle differences between different foreign pathogens that is taken over by adaptive immunity. The components of innate immune system which help in mediating the same include macrophages, neutrophils, physical barriers such as the skin, and many antimicrobial compounds which are synthesized by the host. All of them, they play very important roles. So one thing that might come into your mind is that why are we calling skin as part of the innate immune system? But the first barrier, the first physical barrier to any sort of infection or any immunocompromisation is the skin. The only time you actually have to fear some sort of superficial infection is when there is a wound within the skin, and that also is tackled immediately by by eliciting an inflammatory response, which brings immune cells to that site wherever the wound has occurred. For example, you get a cut. That cut is immediately within a few minutes or a few hours is taken over, is completely covered by cells, so that there is no blood loss, there is no space for any pathogens to grow, it does not get septic, and it does not eventually cause a very severe disease. So that is why the innate immune system, within the innate immune system, the physical barrier of the skin is considered the first line of defense. Along with that, internal mechanisms are also present. There are antimicrobial compounds, there are perforins, granzymes, all of these come in. There are macrophages, neutrophils, etc., which are constantly moving around or circulating within our system, within our lymph nodes at different sites within the body, so that they can constantly monitor whether there is a pathogen or not. In fact, some sort of pathogens, they're also called as some sort of macrophages are also called as sentinel cells, because they are sort of like bodyguards that are constantly present to defend any infection that might come in. So that is the basic idea of the innate immunity. Don't worry, we will be taking up innate immunity in a separate, I think there's an entire week dedicated to innate and adaptive immunity in extensive detail. So this week is more about the overview. Again, if there is any doubt, please bring it up. We will look at any topics if there are necessary in more detail. Then talking about adaptive immunity and the distinction from innate immunity for the same. Adaptive, as the name very obviously indicates, is not something that is present by birth. It is coming up because your body is adapting to a particular intrusion or a particular problem that it has suddenly encountered. That problem is obviously in response to an infection. That infection can either be bacterial, it can be parasitic, it can be viral, it can be a cancer cell as well. Now, cancer cells are a more complicated concept. We will get to that later. It's because cancer does not come about externally, except for a few cases when certain viruses are able to cause cancer. Only in that case, that is an external source. Otherwise, cancer develops within the body by itself. When it does so, that infection is a lot more difficult to tackle, and that is why cancer progresses so rapidly and in such a large percentage of the population as well. So that's a more evolved concept. We can tackle it towards the fag end of the course if anybody is interested. So coming back to adaptive immunity, It is developing in response to an infection. The starting of adaptive immunity is a lot later. Like we said, innate immunity kicks in within a few hours. 4 to 5 hours is the usual response time of innate immunity to tackle the response to a pathogen. However, adaptive immunity begins 5 to 6 days after the initial infection. If the pathogen is able to bypass innate immunity, then only comes in the role of adaptive immunity, and therefore it's considered as the second line of defense. But despite the fact that it is kicking in a much later, it is much more evolved. When we say evolved, it means that the repertoire that exists for conducting adaptive immunity is a lot more elaborate. There are a huge number of cells. There are multiple pathways which are all converging into the branch of adaptive immunity, and that is why most of us are fit most of the time, and only very rarely, when all of these immune defenses have been compromised, does a person not recover from an infection. So it is a lot more evolved, and therefore is a lot more complicated as well, which is obviously something you'll study in a few weeks. It is able to recognize, eliminate, and also remember the invading pathogen. This is what is something we have already stressed upon, that it is able to remember the pathogen that has come in once, and therefore is able to tackle it in the long run. So that's why it said that people who suffer from chickenpox once, usually, it's again not something that can be said with absolute confidence, but usually do not suffer from the same again, unless there's a huge gap, and the virus that had initially infected that person versus that virus which infects them, say 30-40 years later, is very, very different. It has evolved a lot. Then in such cases, an immune system might not be able to fight off that new viral load, and then that person suffers from that disease. So that's a slightly different concept, but usually the adaptive immune system would be able to remember the invading pathogen that has come in. It elicits a memory response in case of future infection by the same pathogen, and as a result, that response is rapid and it is heightened. Rapid as in the time taken to tackle that infection the second time would be much higher. If you remember that graph, we saw that the response time had decreased significantly. So the first response took off a lot slower, versus the secondary immune response is quick, it's steep, it's quick, and it's way more heightened. The antibody concentration is a lot higher. So that is the memory response that is part of adaptive immunity. This memory response is obviously quicker. Like I said, it is stronger, and therefore is a lot more effective in neutralizing and clearing off the pathogen in due course of time. The major components that mediate adaptive immunity are the lymphocytes and the antibodies that they produce. Now, this is something which we can get into a little more detail about. So when we say lymphocytes and the antibodies they produce, what does it mean? It means that the adaptive branch of immunity can also be further classified into two subclasses. When we say subclasses, it means that they do not operate separately. Both of them are in action when adaptive immunity is kicking in. They eventually merge together, and we'll see how they merge together as well. But the two branches or the two subclasses that we can now refer to are the humoral branch of immunity and the cell-mediated immunity. Abbreviated, it is written as HMI, which is humoral mediated immunity, which is humors basically mean antibodies, and CMI, or cell-mediated immunity, is taken care of by lymphocytes. And in this case, those lymphocytes would be T lymphocytes. We'll come into detail about that as well. So these are the key differences between innate as well as adaptive immunity. Like I mentioned, when we talk about components of the immune system, I specifically talked about the skin. Here you can find that in a little more detail. Also for reference, most of the material that I will be showing in my slides are from Kobe Immunology. As Professor Ghosh had referred in the lectures as well, you can refer to any textbook. He had mentioned 4 textbooks. You can go through any of them. If you can find online PDFs, or if you have access to a library, you can get these. So, but I would very, very strongly recommend that you go through a book, because immunology, while is extremely interesting, there is a lot that comes under the branch of immunology, and lots of things merge together to eventually formulate the immune system. So understanding that a little comprehensively would be helpful for you, and that can be done with the help of a textbook. So Kumbhi immunology these are all from a slightly older edition. This is the 5th edition, which is what I had on me. But if you have access to the 8th or 9th edition, please go ahead and look into it. It's a wonderful book, and it'll clear up your concepts very easily. So here what we see in on the left, the table here shows a summary of the non-specific host defenses that are present. Why is it called non-specific? That's because it is part of the innate immune system, like I discussed. So when it is innate, it is not responding to a particular pathogen. It is just responding to the fact that there is some intruder, somebody who was not supposed to be there is now within our system, and we need to clear it off as soon as possible. So as part of that system, we look at different types of barriers. Again, these will be discussed in more details in the upcoming lectures. I just wanted to familiarize you with the concept, which is why I've included it here. So the first subtype under the non-specific host defense looks at anatomic barriers. As you obviously understand, anatomic means something which is physically present, which is part of the human anatomy. And which is how do I put it? It's not a molecular mechanism. It's something that you can touch and feel, and you can visualize very easily. So under those two, and under the anatomic barriers, the two subtypes are skin and the mucous membrane. I think somebody is unmuted probably. If there's a question, please interrupt me. So under anatomic barriers, the first one is the skin, like I said, which serves as the mechanical barrier against the microbe that could invade our system. Secondly, it's not just a mechanical barrier. There is also a pH which is maintained approximately between 3 to 5, which retards the growth of microbes. So why this is also why it's important to be an informed scientist, to be an informed student, is because what you see commercially as products which are sold to be good for the skin, they often go for pH which is higher than this. Neutral pH is usually broadcasted as the more as a better product, but that does not necessarily mean the same. A pH of 7 is something which will not really help tackle bacteria, or will not necessarily clean off any microbes that might be present. Instead, a slightly acidic pH, which is within the range of 3 to 5, is better. It will allow less microbial buildup. It will lead to less infection, and therefore is more pH balanced as in contrast to what you can commercially see. So leaving the skin alone would be much better. It will serve as a much better immune system and can do a good job. Secondly, mucous membranes. Now, while skin is something that you obviously can see externally, the mucous membranes line the inner side of the system of any individual system, and they contain apart from serving as just the mechanical barrier, they also contain microflora within them. Microflora means the environment that consists of a diversity of microbes that are healthy and helpful microbes within the system, and which are necessary for tackling of pathogens which can cause disease. So I'm assuming that you might have the background in microbiology, that not every microbe is harmful. Only those which are disease causing are pathogens. Those which are just harmless, which are able to either serve as commensals, which means that they are able to contribute something to the human system and derive some help from the immune system, they are very helpful. For example, the gut microbiota that is present, they line the mucous membrane of the entire gut, and they are crucial for us to be able to digest food and to be able to tackle most of the pathogens that might come as part of the food that we consume regularly. So mucous membranes consist of normal flora that competes with other microbes for attachment sites and nutrients. The mucus is, as you can understand, is a very slimy, sticky sort of layer that is present towards the inner side of this lining, and that is able to trap any foreign microbes that might be present. And also the mucous membranes are usually lined with cilia. You might be familiar with it. For example, if you had studied the digestive system, then as part of the small intestine, we had learned that there are villi which are present throughout the intestine, which are like very small microscopic projections which help in better absorption of nutrients. Similar to that, cilia are present throughout our, for example, the nasal tract, for example, is lined with cilia, which helps in propelling microbes out of the body. So, for example, when you inhale, you are inhaling not just air, but somebody's mic is muted. Okay, so you inhale not just air, but also a wide range of microbes. So to tackle that, these are physical barriers that are present. Apart from that, there are physiological barriers as well. This person. So there are physiological barriers as well. That includes temperature, low pH, and chemical mediators. Temperature, as in the normal body temperature, inhibits the growth of certain pathogens, which is somewhere around 37 degrees most of the time. And fever response instead inhibits the growth of pathogens. So most of the time when you get a fever, whether it's a viral fever or it's because of some bacterial infection, your body, or in fact your hypothalamus within the brain, which serves as sort of like the thermometer of individuals, of humans, that purposely raises the temperature of people. Just So that particularly raises the temperature of such individuals, and overall increase in temperature eliminates a large number of bacteria because they are no longer able to survive at those higher temperatures. So that is a physiological barrier. Secondly, low pH, like for example, the acidic pH of the stomach is helpful for killing off most microorganisms that are not a part of the gut microflora. Chemical mediators, for example, lysozyme. Lysozyme, most commonly we study that it is present in our tears. So it is a mechanism of clearing off pathogens from the eye so that you don't usually get eye infections. It is only when a lot of the immunity within the eye is compromised, only then such people would encounter eye infections. Interferons, which are extremely crucial for antiviral response mechanism, they are present in uninfected cells, and that is the reason why most of the time not every virus is able to cause a disease within us. Only sometimes it does happen when the immune system is not able to tackle it. The complement system, I'm not getting into the details of it because the complement is a elaborate concept. It's a cascade. There's a system where multiple molecules they work one after the other in order to mediate an immune response. It is a part of the innate immune system, not adaptive immune system. Please keep that in mind. It lyses microbes and it facilitates phagocytosis. Then there are TLRs, or toll-like receptors, which recognize microbes. They signal the cell to secrete certain immunostimulatory cytokines. And there are collectins, which disrupt the cell wall of pathogen. Don't worry, each of these components will be discussed as part of the innate immunity lectures. There are also phagocytic or endocytic barriers where cells internalize or endocytose microbes, break them down, and then flush it out of the system. And inflammatory barriers, like I just mentioned, that when you get a cut on your body, physically a cut that is exposing the soft tissues of the body, then there is a number of mechanisms come into play. There is edema, erythema. Edema means there is redness. Erythema means there's swelling, and it brings a large number of immune cells at that particular site so that you don't suffer from any disease because of that exposed skin. So all of these are non-specific defenses and they form a part of the innate immune system. Now, moving ahead from that, the second branch that we discussed is the adaptive immune branch. Now, adaptive immunity, like we said, is a lot more complex, a lot more evolved, and a lot more specific. There are four key features of adaptive immunity. Keep this in mind. We talked about initially the features of just immune system at large. Innate immunity was a little more non-specific. It had a more preliminary responsibility. But adaptive immunity has four very, very key features. These will also be discussed in more details, but the features are A, antigenic specificity. The importance of this is something you'll understand as we go ahead with the course, that responds to one particular antigen, or in fact, you will get into more detail about it. One region of one particular antigen is extremely important and extremely specific. Second comes diversity. The adaptive immunity is not choosy in the sense that it will only respond to one particular type of pathogens. No, it can tackle any and every pathogen or any disease-causing organism that is present within the system, like around us in the surroundings. Third, immunologic memory. We have discussed this multiple times. That is, it is able to remember and it is able to retain information that it has once encountered, and therefore is able to tackle that infection much better. And fourth comes self, not self recognition, which is something we've already discussed previously. So these are the four main features. What you see diagrammatically represented below is actually a part of the phagocytic endocytic barrier that I just mentioned. It has been schematically represented here. So what you see here in green is just a schematic representation of a bacterial. For example, some pathogenic bacteria, let's say E. coli, for example, has entered our system somehow from some food that we consumed, or from water, or just because of touching and then consuming something from there. So the bacterium is recognized. It attaches to certain membrane invaginations called as pseudopodia. Pseudopodia is something I assume you must have studied before also as a part of amoeba. So pseudopodia are temporary or transient sort of projections which come out from cell. They help in surrounding a pathogen, or in some cases even surrounding food items. So just for taking something up into the cell that is carrying out this activity. So these pink projections you see, these pseudopodias, they actually surround the bacterial system. This bacteria is completely ingested by that particular cell, and it forms something called as the phagosome. Phagosome simply means an enclosure or a sort of sac-like structure which contains the material that has been phagocytosed. This phagosome then fuses with the lysosome, and this forms the phagolysosome. So what you see here on the left is another entity which has you can see these little blue dots. This is representing a lysosome, which is obviously an organelle, cell organelle, which contains a very high titer of enzymes which help in breaking down anything that enters the cell, or for digesting anything that enters the cell. So this lysosome and phagosome, they fuse, they form a phagolysosome. This phagolysosome is then able to carry out the digestion of that particular pathogen, of that bacterium that had entered. It completely digests that material, and then those digestion products are released from the cell. So this part is partially under the phagocytic barrier of innate immunity. Now, what is being released here? Those little components that have been released here will be taken over by adaptive immunity to remember or to retain information that if in case this same bacteria enters the cell, enters the individual system sometime in the future, then the immune response can be elicited very quickly. So you see how these two branches they merge and they mediate immunity as we know it today. So just a second. Yeah. So these were the components of innate and adaptive immunity. Adaptive immunity we just mentioned very briefly. Now coming to the main components that are a part of the adaptive immunity. We said that lymphocytes and their secreted products, in this case, those are antibodies, they mediate the adaptive branch of immunity. So what you see represented here are the three major types of cells, which are called as lymphocytes. Where the name comes from is something we'll look at in the next slide. So lymphocytes are of two types, majorly of two types, B cells and T cells. So what you see here, the first schematic is representing a B cell. And in the figure legend, you can see that B cells have approximately 10 to the power 5 molecules of membrane-bound antibodies per cell. That's around 1 lakh antibodies on just one cell. Now imagine the number of cells that can circulate in a system when there is an active immune response that is going around in response to a pathogen. So there's a huge repertoire of antibodies which are present, and they are able to completely surround that pathogen which has come in, and they're able to eliminate the same. So all the antibody molecules on one particular B cell have the same antigenic specificity and can interact directly with the antigen. So this is what you see here represented, this Y-shaped structure, if that is obviously the antibody, and this particular region at the top, the top parts of that Y-shaped structure is exactly where the antibody binds, where the antigen binds. Now, you might have this confusion that do we call a bacteria, a particular bacterial cell as an antigen? No. A bacterial cell is a pathogen. One particular portion of that bacterial cell surface which can be accessed by this antibody is an antigen. It's a very small part of the entire pathogen that has come in. So for one particular bacterial cell, there can be hundreds and thousands of antigens that can be derived as a result of the bacterial cell entering the body. Now, each of those can all act together and therefore eliminate that bacterial cell from that system. If it is not entirely clear, I can try explaining it better, but once you understand over time, you'd be able to understand it in a little more detail. So these are B cells. What you see here represented in blue and pink. Okay, in blue and pink, these are two types of T cells. So B cells, as you can see, these are responsible for producing antibodies on their surface, but T cells do not have the same. T cells are generated from they mature in the thymus, and they are of two major types, Th and Tc. Th is for T helper cells, or Tc are for T cytotoxic cells. So when we say helper T cells, what do they help in? You'll learn about it in much more detail later. T cytotoxic, as the name indicates, that it is causing cytotoxicity, or it is killing cells basically. So on the surface of T cells, what we see are these receptors, TCRs or T cell receptors. In fact, antibodies present on the surface of B cells are BCRs or B cell receptors, and they are otherwise also called as antibodies. But T cell receptors are not the same. They are present on the surface. But what do you see as a difference between TH and TC cells are these pink and these blue colored molecules. These are called as CD. These are also antigens present on the surface. They are cell surface receptors, and these are called as CD. CD stands for cluster of differentiation. So CD4 is one particular type which is present on T helper cells, and CD8 is of a different cluster of differentiation, and it is present on TC cells. So these are how you distinguish these different types of cells. So these are called CD4 plus, these are called CD8 plus. T cells that bear CD4 or CD4 positive cells recognize only antigens that are bound to class 2 MHC molecules. We'll come to this. T cells bearing CD8 or CD8 positive T cells are able to only recognize antigens that are associated with class 1 MHC molecules. In general, CD4 act as helper T cells, CD8 act as cytotoxic T cells. Both types would be able to express approximately 10 to the power 5, once again around 1 lakh cells. 1 lakh identical molecules of the antigen binding TCR per cell, all of them having the same antigenic specificity. So one cell, one T cell will produce antibody or will be specific for only one antigen. There can be a population of T cells that will all be specific to one pathogen, but each of them can have different specificities. But on one cell, all of them, all of the antigens which are present, all of the TCRs which are present are specific for one antigen. If it is confusing, please let me know. I'll try to explain it a little better. So these are the different subtypes which are present within the adaptive branch of the immune system. We'll then come to what exactly happens there. The four related but distinct cell membrane molecules that are responsible for antigen recognition by the immune system are, First of all, like we just saw, the membrane-bound antibodies that are present on the surface of B cells. Second come T cell receptors. T cell receptors we saw are TCRs, which each of which have the same antigenic specificity on one particular T cell. Third are class 1 MHC molecules. Fourth are class 2 MHC molecules. Each of these molecules plays a very unique role in recognizing antigens, and it ensures that the immune system can recognize and respond to all of the different types of antigens that it encounters. So that is the job. That is why we call the adaptive immune system as diverse, because it is creating an entire army of T cells which can tackle different regions of the same pathogen and altogether mediate the immune response smoothly and very accurately. So this is how it works. Now, what you see on the right is a representation of how these different types of cells are generated, what starts it off, what are the different subtypes, and what are basically the components of the immune system. So the key cell, the headmaster at the top, is the hematopoietic stem cell, or abbreviated, it is called as the HSC. Hematopoietic stem cell, where is it present? It is present majorly within the bone marrow. That is where stem cells are present. They are present in a very protected environment. They are self-renewing. If you have come across stem cells before, you would know that it is an intrinsic property of stem cells to be able to self-renew. When we say self-renew, it means that they're able to replenish, they are able to regenerate themselves. And from that stem cell, they can then differentiate into more specific lineages or more specific subtypes of cells that would eventually end up performing different types of functions. So from the hematopoietic stem cell, what you see are the derivatives of two different types. One is a myeloid progenitor, second is a lymphoid progenitor. Let us first look at the myeloid progenitor. From the myeloid progenitor, there can either be direct development of the dendritic cells. So dendritic cells actually have a very important role to play as well. If you had listened to the lectures carefully, you would know they have a very important role to play. I'll not mention it directly, but we'll come to it somewhere around the end of the class in case you don't remember. So dendritic cells are one particular subtype. Second, what you see, the myeloid progenitor can then differentiate into several other subprogenitors. Progenitors, as the name indicates, it is something which is primitive to the actual state. So it's a precursor. It's a precursor cell that can then further differentiate and become a more specific cell type. So from the initial stem cell, you get myeloid progenitor, and from there you can get other subtypes of progenitors, among which the first one you see here is the granulocyte monocyte progenitor, which is a precursor cell that can either differentiate into a monocyte or it can differentiate into a neutrophil as well. So if it, in case it differentiates into a monocyte, that monocyte can further go and differentiate into macrophages. Monocyte macrophage differentiation is something you'll also study eventually. Otherwise, the same progenitor can also differentiate into a neutrophil that is present within the system. Second comes the eosinophil progenitor. Eosinophils, if you have studied it before. So the nomenclature of these different cell types has actually come about because of the way in which they get stained. So eosinophils, basophils, and neutrophils, they stain differently because of the difference in pH that exists. This is something you can look into. It's a very simple and very interesting concept as well. And they perform different types of functions. So from a blood smear, in case you have done this experiment as part of your practicals, if you just take one droplet of blood and then you smear it on a slide and you observe it under the microscope, of course, the major population would be your RBCs or red blood cells. But apart from that, all of these WBCs or white blood cells, which are part of the immune system, can be distinguished based on the way in which they get stained. So the eosinophil, in this case, eosinophil progenitor gives rise to the eosinophil, majorly that is responsible for tackling parasitic infections. That is when it becomes very high. And then you have the basophil progenitor. Apart from that, there is the formation of a megakaryocyte, which is the precursor towards platelets. You must be familiar with platelets by now. And of course, the erythroid progenitor, which gives rise to erythrocytes or RBCs. So from the initial myeloid progenitor, you are getting RBCs, WBCs, as well as the dendritic cells, all of these different subtypes. On the other hand, In case the hematopoietic stem cell differentiates into a lymphoid progenitor, then the situation is a lot different. Lymphoid progenitor, as the name indicates, will eventually give rise to lymphocytes, right? So either it can give rise to lymphocyte, or on the other hand, it can directly differentiate to form an NK cell or a natural killer cell. If not, then it can either differentiate into a T cell progenitor or a B cell progenitor. From the B cell progenitor, it will of course form the B cell. From the T cell progenitor, it will form the T cytotoxic that we just looked at, the CD8 plus T cells, or the T helper, CD4 positive T helper cells. Interestingly, what I just mentioned, you can see that dendritic cells can also be derived from the lymphoid progenitor. There has been some update on this in literature as well. You can look it up if it is interesting. If you don't find it, we can discuss it in the next class. But you see that dendritic cells are also actually present in both branches of this of the immune system. So from the main master, you see lymphoid derivatives and you see myeloid derivatives all together, which form a part of the cell-mediated branch of immunity. So this is what we see here. Now, what you would what you should keep in mind is that apart from the cell-mediated branch of immunity, where all of them are operational, there is one particular subclass within these also. That subclass is of antigen-presenting cells. What does it mean? The name is it's direct. Antigen presenting cells simply mean these cells are able to present or display some antigen on their surface. Why can't every cell do it? That is because if in case it was able to do so, then there would be a lot more non-specificity. Any cell would be able to display any pathogen and there might be inaccuracy there. It would overburden the immune system, and a hyperimmune response is extremely dangerous and almost always very difficult to tackle and can also become lethal in some cases. So therefore, controlled and regulated immune response is extremely critical for that matter. So among this entire population of all of these different types of cells, the antigen presenting cells, otherwise known as APCs, include three subtypes. First of all, dendritic cells, B cells, and macrophages. These are the three subtypes that are mainly responsible for presenting antigens that will be recognized by MHC molecules and that will eventually be degraded from the system. How that happens is something we'll come to. So I hope everybody is able to understand things. If not, again, please interrupt me. Just wrong here. Yeah. So like I had mentioned previously, again, these schematics are all from Kubi Immunology. You can look at it. You can look at any other textbook also. You can just search online as well. You'll find very nice, very well updated review papers also, which explain these concepts in great detail. Now, continuing with the idea of the adaptive immunity, and within that the humoral and cell-mediated immunity, we will look at these two branches independently. First, let us look on the right. Okay, no, let's start from cell-mediated immunity itself. What you see at the top are representations of different types of pathogens. So either there can be some foreign protein. Why are we just talking about foreign protein? It might be so that an entire pathogen does not enter your system. It just releases some protein and that is enough to cause disease. In fact, in case you are not already familiar with it, you can look at the concept of prions, which are called as proteinaceous infectious agents. So they are simply protein molecules that are able to cause disease within humans. So that is one possible pathogen as well. So foreign proteins, viruses, bacteria, parasites, fungi, all of these can present antigens to a particular individual. So when this is where you see step one, when there is an antigen entry into a system, that internalized antigen, what you see represented here, gets digested by a particular cell. So once this antigen has entered, it is getting digested, which is why you see the borders are sort of serrated. As a result, what you see, there is a altered cell, which is able to present that antigen. If it is presented by, it can be So what you need to understand here, like we mentioned previously in this slide, yeah, here you saw that the T cells which carry CD4 and CD8 are able to present cells in different ways, or they are able to recognize cells in different ways. How? T cells that carry these CD4 positive cells or the T helper cells are only able to recognize those antigens which are being presented by MHC2 molecules. What is MHC? MHC is major histocompatibility complex. It's a cell surface receptors. Let us just understand that for now. So MHCs can be of two subtypes. If you had gone through the lecture, you will see that there are two types, MHC1 and MHC2. Their structure and their antigen binding region, all of these are different. And among them, the T helper cells are only able to recognize antigens that are bound to class 2 MHC molecules. On the other hand, CD8 positive T cells are able to only recognize those which are presented on class 1 MHC molecules. Keeping this in mind, if we come back to the schematic, we see that these cells, again represented here as T helper and TC cells, are able to recognize certain antigens here represented in red, presented by these two different types of MHC. When it is presented by class 2 MHC, you see that helper T cell or CD4 positive T cell is able to recognize the same. On the other hand, when it is being presented by class 1 MHC, it is being recognized by TC cells or T cytotoxic cells. So once this recognition is occurring, that recognition involves interaction between the MHC and the antigen receptor present on B cells, dendritic cells or macrophages, which are antigen presenting cells. And once the T cell receptors have recognized this particular antigen, then there is further co-stimulation by the CD4 or CD8 co-receptor present on the cell surface, and then it allows the selection of that particular T cell. So basically within a system, you would have n number of T cells which are constantly circulating. If a particular T cell is able to encounter an antigen presenting cell that is presenting its specific antigen, that means that within that individual, there is an invasion of a pathogen. Say, for example, let's say there is uh, say for example, I have like thousands of T cells, which are thousands, millions of T cells that are circulating within my body. Now I am suddenly encountering, for example, coronavirus. Coronavirus enters my system. So from there, the T cells, the subpopulation of T cells, which are capable of recognizing some part of SARS-CoV-2 will be activated. They will be able to bind to the B cells, dendritic cells and macrophages, which are presenting some part of coronavirus, and they would be able to recognize the same. Once they're able to recognize, these individual cells will be selected for. What does it mean selected for? As in that this particular cell will now duplicate and rapidly increase in number, so that the number of cells that are able to recognize, identify and recognize the invasion of SARS-CoV-2 within my body increases very suddenly. That leads to formation of activated T helper cells. So these activated T cells, whether it is TH or TC, depending upon the type of initial pathogen that had come in, would then be able to secrete cytokines that contribute to the further activation of B cells, TC cells, and other cells. So T helper cells by themselves directly do not really do anything. As in they do not directly cause killing of cells. They work as helpers, and they activate pathways that will help in clearing out that infection in other methods. So for example, it will activate B cells. Which B cells? B cells that are specific for that particular antigen, or in this case SARS-CoV-2. So that will produce antibodies which can bind to SARS-CoV-2 and either neutralize it or eliminate it from my system. So that is how the two branches of immunity are connected to each other. So this becomes a part of the cell-mediated immune response. On the other hand, these cytokines that were released as part of the cell-mediated immune response can trigger the second branch of immunity, which is the humoral response, which is what I just mentioned, as is shown on the right, to look for those B cells which have encountered their antigen. And these B cells will also now be activated, and the activated B cells are called as plasma cells. Otherwise, you can see they're called as antibody secreting plasma cells. So these cells are increasing in number. what happens upon selection of these B cells is that plasma cells produce the same receptor, produce the same B cell receptor, but they are not membrane bound. Instead, they are secretory in nature. Why is that important? The initial B cell that was present had the antibodies on its surface so that it could bind and it could recognize that this particular antigen has now come in. Once that recognition part is complete, now we move on to effector response. To mediate that effector response, that B cell now has to synthesize antibodies which can be released into circulation. And these, when I say circulation, they have to go throughout the entire body, because it is not necessary that coronavirus has just invaded one part of my body. It could be that it started off from the nasal tract but has spread throughout the system. So to carry out a systemic monitoring, a huge number of antibodies are required, and therefore the secretion or the release of these TCRs, of these BCRs or antibodies becomes extremely crucial. So these antibody secreting plasma cells will now take over the job of increasing the antibody titer of such individuals. Then that antibody will go throughout the circulation of that affected patient and will facilitate clearance of that pathogen from the body. So this is how the humoral and cell-mediated branch of immunity are connected to each other. Let's just read the legend once so that we can assimilate everything that we have said till now. So this schematic is providing you an overview of the humoral and cell-mediated branches of the immune system. B cells first are able to interact with the antigen and then differentiate into antibody secreting plasma cells. The secreted antibody can then bind to its antigen and facilitate its clearance from the body. On the other hand, in the cell-mediated branch of immunity or cell-mediated response, various subpopulations of T cells can recognize antigen presented on cells. T helper cells will then respond by producing some cytokines. TC cells will respond by developing cytotoxic T lymphocytes or CTLs, and those will mediate killing of altered cells, whether it is virus infected, whether it is some other pathogen that has come in, etc. So this is how the two branches are connected. These might be very new concepts. Don't be afraid, you will be able to understand everything. If not, go back to textbooks. If it still doesn't help, I'll get better, more detailed schematics to explain the same. But yeah, you have to come up and you have to tell me where you're facing difficulty. Do not hesitate. Everybody's here to learn, including me. I am also learning a lot of concepts so that I can help you guys out as well. So please bring up any questions if you have in mind or any topic that you just want to be discussed. So so we talked about how the humoral and cell-mediated branches of immunity are connected to each other. Now comes the next part, that how is that selection part being carried out? On the left, first, let us focus at the schematic on the left. What you see here is the initial hematopoietic stem cell, which is present within the bone marrow. It is leading to the formation of certain types of B cells. Let's not get into detail about gene rearrangement. It's a very complicated concept. It's the one I just told you. Susumoto Onegawa was awarded the Nobel Prize for it, for the way in which gene rearrangement is carried out for antibody production. It's a fantastic concept, just a little difficult to understand. So this gene rearrangement is giving rise. So different combinations of rearrangements are occurring, and that is leading to the formation of mature B cells of different types. Here it is labeled as 1, 2, 3, and 4. So they all are carrying antibodies on their surface. Approximately, we saw that around 10 to the power 5 antibodies per cell, or antibodies or BCRs per cell. However, what you need to very clearly understand is that between these different subtypes, that is B1, B2, B3, and B4, they are all containing antibodies with different specificities. When I say specificity, it means it is binding to a different antigenic epitope. What is an epitope? A little complicated concept, but epitope is the smallest part of an antigen to which an antibody binds. Say for example, let's consider that this is a particular antigen, right? Now you have different populations of antibodies. One antibody will bind here, another can bind here, another binds here. They're all targeting the same antigen, but different regions of that same antigen. So this particular region is one epitope, this is another epitope, this is another epitope. So antigenic epitopes can differ, but eventually that entire population of antibodies is targeting the same antigen. Why is this necessary? Why could we just not have one antibody which is binding to just one site, and that could just have been enough? That you produce hundreds of thousands, millions of cells that produce that antibody and then just clear that out from the system. Why are we not just doing that? A, because that would slow down the response significantly. When you have a multifaceted or multi-pronged approach, you will find different ways in which you can target the same antigen. Secondly, it might so happen that while this, for example, the first time I'm getting infected by coronavirus, this particular epitope is exposed. This is where my antibody is binding and it is eliminating this pathogen. It might happen that the next time I'm facing the same infection, this epitope has changed. Somehow, some amino acids have been changed, or this epitope or that protein has folded differently. It has now folded like this. So that epitope is somewhere hidden. As a result, antibodies are no longer able to bind it, and therefore they're not able to clear off that infection, and therefore that person suffers. So to bypass that, to utilize energy in the most resourceful way, most judicious way, when a particular antigen has been identified, for when a particular pathogen and an antigenic epitope on that pathogen has been identified, a lot of resource is dedicated towards increasing that in number. Now, from this particular schematic, however, the idea that they are trying to convey is different. They are looking at one particular subtype and they're able to recognize only one antigen which is able to bind to that pink colored cell, which is your the B cell of type 2. The rest of the cells are not able to bind to any portion of the antigen. As a result, it is now extremely important to carry out the selection of only this particular B cell and not the other cells. Also, what you should keep in mind is that the location is important for clonal selection. So you see that the initial population is present within the bone marrow where there's the stem cell, gene rearrangement is occurring, and all of the different types of B cells can be produced. Then maturation into antigenically committed B cells. Antigenically committed as in they now have specificity for one particular antigen and they're committed towards only that particular antigen. Till here, you see the location is the bone marrow. Now, once they have moved out of the bone marrow and they have lodged into the peripheral lymphoid tissue, that is when there is a possibility of encountering an antigen, because usually antigens would obviously not be present within the bone marrow. If that is the case, it's a very serious condition where that person might not necessarily survive. But instead, what happens, these sentinel cells or these bodyguard cells basically, they move from that from the site of the bone marrow towards peripheral lymphoid tissue within lymph nodes and other lymphoid organs, which is something you already came across in the lectures. And there they might or might not encounter the antigen. In this case, from that entire population, only antigen 2 specific B cells are able to encounter their antigen. If that happens, when there is an antigen antibody interaction or antigen antibody recognition that has been facilitated, then there is an antigen dependent proliferation and differentiation of that particular B cell. So now this B cell gets selected, clonally selected, and there is clonal expansion of this particular cell in huge numbers. How is that done? From type 2, you see that cells are increasing in number, and from there, one subpopulation remains as memory cells so that the system can retain information that there was infection via this pathogen, and the rest of the subpopulation is able to divide into plasma cells, which is what you see are much larger in size. They have all of the organelles, etc. And what they do is they produce antibodies which can be secreted. So both memory cells and plasma cells are produced, from which you get a very high antibody titer. So antigen dependent proliferation and differentiation into plasma and memory cells is carried out in the peripheral lymphoid tissue. Okay, so this was the idea of clonal selection. Just give me a minute. we don't have a lot of concept left, so don't worry. We'll discuss a few, few sample questions, just a few more topics and a few sample questions, and then we can get to your doubts. Okay. So, after clonal selection, the next concept that we can look at, it has already been explained before, but I found some good schematics, so I thought I'll bring it up also, is the idea of antigen processing. So what you see at the top is a condition where there is an antigen which is exogenous in nature and which has been ingested or taken up by a cell through a process known as endocytosis or phagocytosis. So once that has happened, using those pink colored pseudopodia, as we had already seen before, that pathogen has been taken up. It is then encountering the endosome that is present. It is forming an endosome. Within the endosome, that particular antigen is being broken down or is being chopped up into smaller pieces. It is encountering the lysosome, which is already an organelle that is present within the cell. And this pathway by itself is called as the endocytic processing pathway. When we say endocytic, it means the entire process is taking place inside the cell, within the cell interior. Now, this particular endosome and lysosome will fuse. Endolysosome formation takes place. Now, from the intact antigen, the antigenic peptides or the antigenic fragments that have been generated will then bind to class 2 MHC. And upon binding, they are MHC, as you see here, are okay, the structure of it is something you'll learn later. But right now here you can see that class 2 MHC has these two sort of independent entities which are together binding or holding onto the antigenic peptide. And then that is being exposed onto the surface. And now this particular cell will serve as an antigen presenting cell. Why? Because it is presenting a part of that antigen on its surface as a peptide and class 2 MHC complex. So this entire process is known as the endocytic processing pathway. On the other hand, if we look at a different scenario where the infection is not in this manner, but it is leading to the formation of an altered cell. Good. So what you see here is that there is the infection by a virus shown here in green, where upon viral infection, if you're familiar with it, then viral DNA or viral genome is injected, which gets converted into viral mRNA and eventually the viral protein as well. Eventually the viral protein as well. And then this viral protein then is taken up by the system. It is being digested by via the vesicular trafficking and the endocytic pathway. And then it is presented on the cell surface along with class 1 MHC molecule because it's a viral peptide. So this forms a peptide class 1 MHC complex. So different types of pathogens are being processed in different ways and are being presented with different types of MHCs. Why is that crucial? Because based on the type of MHC which is presenting a particular pathogen, recognition by different T cells will be carried out and that pathogen will be dealt with in a different manner. So that is the idea we get from here. So yeah, so till now, what did we see? We first understood the basics of the immune system, its characteristic feature, the innate immunity and adaptive immunity, the differences between the two, how it works as the first line of defense and the second line of defense, what are the components that form a part of the innate and adaptive immune response. What happens when innate immune response is not sufficient? How does adaptive immunity take in? What are the different subcomponents or the sub branches that are present within adaptive immunity, which were humoral and cell-mediated immune response? What allows humoral immunity? It is majorly B cells. What allows adaptive immunity? It is majorly by the T cells. Neutrophils, macrophages, etc. are also present. How the complement system is part of the innate immune system, how the cell-mediated and innate immune, cell-mediated and humoral immunity branches are linked to each other. Which cells comprise the same? Why is it necessary? What are its characteristic features? What are the different types of cell surface molecules that are present as part of the adaptive immune system, and how does it regulate the same? The effector mechanisms have already been discussed in the lectures, which is why I did not include the same. it would be just a repetition of the same, so I did not include that. So keeping in mind this entire overview of the innate and adaptive immune system, it depicts a very interactive multi-component system that protects the host from infectious diseases and also from cancer. Again, the protection from cancer is a very it's an advanced concept. We are not unnecessarily getting into that detail right now. But yes, it is extremely important for protection against cancer as well. Now, sometimes the immune system can fail to protect the host adequately, or it can misdirect its activities, which cause discomfort, sometimes some debilitating diseases, or even in the worst case scenario, it even causes death. So while the immune system is extremely crucial, if it goes awry, it can wreak havoc completely. Now, the most common manifestations of immune dysfunction are as follows. First, allergy and asthma, which is something you find very commonly among people around you. Second comes graft rejection and graft versus host disease. This is very, very, very crucial. So graft rejection is a very common, it's a concept that has now been sorted to a large extent, because initially people only used to do blood type matching that I mentioned was performed by Karl Landsteiner, that if somebody has, is A positive or AB positive for that matter, and that person is just given another AB positive person's organ, people used to think that would be enough. But it turns out that that is not enough. There are more entities that are declaring themselves as non-self or as different from the system in which they have been introduced, and that can cause a lot of problems. So there is another type of matching which is carried out, which is HLA or MHC or HLA type matching, which is human leukocyte antigen matching, which is carried out between an individual who will be a recipient of an organ or a recipient of a graft versus the donor. So that HLA type matching has to be carried out so that the organ which is being transplanted does not get detected as some sort of a invader or some sort of a pathogen, and then it is removed from the system. Also keep in mind that when it is, for example, just a pathogen, a bacterial pathogen or a fungal pathogen even for that matter, that in itself triggers such a huge immune response. In contrast, when it is an entire organ, say for example, a kidney transplant is being carried out without HLA type matching, and that entire kidney is getting detected as a pathogen by the body. In that case, the immune response that is generated is so severe that it's completely, it would be out of control, and such a person is extremely sensitive to any medication. It's very difficult to tackle such a situation. So that is graft rejection, where a particular individual's immune system, it rejects the organ that has come in or the graft that has come in. Versus the other type is graft versus host disease, where the graft which has come in is rejecting the organ, so that is rejecting that particular individual. So it can be both ways, because the graft also comes from with its own very minimal but its own immune system. If that graft that has come in detects the system or that individual as foreign, then it can trigger a very, very, very hyperactive immune reaction. So both these types of rejections are seen. In fact, one of the most, while liver transplants are very not very, but relatively easy to carry out, some very like the large number of reports that show this graft rejection or graft versus host disease are reported mainly for corneal transplants, for people who have compromised eyesight or at some point encountered an accident, etc. Often undergo corneal transplants, but there's a very high rate of rejection in those cases as well. There are success stories also, but rejection is has quite high propensity. So this is a form of immune dysfunction itself, that there can be graft rejection or graft versus host disease. So to prevent that, usually the method of choice is that in hospitals, people who are people that would serve as recipients are usually subjected to very, very high doses of immunosuppressants. So you are shutting down, not completely, but at least dumbing down the immune component of an individual, so that their overall immune response is slower. While that is good for this graft uptake, or helpful at least for the graft uptake, it makes that particular patient very, very, very sensitive to secondary infections within those hospitals. So it's a tricky deal, but yeah, these surgeries are regularly carried out and there are huge success rates as well. So that is about the graft versus host disease or graft rejection. Third are autoimmune diseases. I've already talked a little bit about it. You can look at some examples. Of course, HIV is the most common example which is cited because HIV, the virus, causes AIDS, which is acquired immunodeficiency syndrome. You can study a little bit about SCID, which is severely compromised individuals. And then, in fact, there are diabetes, there are rheumatoid arthritis. These are all examples of autoimmune disorders. And immunodeficiency also becomes a subpart of this type. So these are the most common subtypes of immune dysfunction. I did not get into more detail about this, but this is overall the idea. The consequences are something which you obviously understand. But yeah, the immune system needs to work in a certain way in order to facilitate proper functioning of or proper survival of individuals. So with this, I'm coming towards an end, towards the topics that I wanted to discuss for today. I just have like few 4 or 5 questions as examples. We will not be discussing your assignment questions now. We can discuss them once the deadline is over. Most probably we'll be doing that sometime next week or the week after. We can take up both week 1 and week 2 assignment questions and discuss those. But for now, these are some previous year questions that were available. So you look at the first question where the immune cells that form a bridge between innate and adaptive immune responses are A, macrophages, B cells, dendritic cells, or T cells. So if you remember that schematic where we saw that there are certain cells which are present as part of myeloid as well as lymphoid lineage, it's actually the dendritic cells that do so. In fact, it was mentioned in one of the initial lectures for this week as well. So how do they bridge the innate and adaptive immune response is something I would like you to go back on and study a little bit about it. If not, I'll discuss it in the next class with you. So dendritic cells are the bridge between these two immune systems. Immune responses. Second question is for hematopoietic stem cells, which are pluripotent in nature, which means that they are A, antigen-specific cells, B, they're capable of developing into any of the blood cells, C, they are committed to producing cells of a single lineage, or D, they are not self-renewing. So first of all, hematopoietic stem cells are not antigen-presenting cells that would defy the antigen-specific cells that would defy the idea behind stem cells. They are not self-renewing is also untrue. Of course, stem cells should be self-renewing in nature. Now comes the idea of pluripotent. So there are terminologies. If you're already not familiar with it, we can discuss it. There are certain terminologies which are used for different types of stem cells. So unipotent, multipotent, pluripotent, totipotent. These are all the different types of cells that can exist. You might already be familiar with it. You can brush up on it again. So unipotent means cells which are differentiated and are of only one particular type. They are completely differentiated cells. Or they might, you can even think of some progenitors. For example, the megakaryocyte, for example, is unipotent in nature, that it will only differentiate and give rise to platelets. So they are precursors, but they cannot differentiate into different subtypes. So those are unipotent cells. Second are multipotent cells. Multipotent cells can produce cells of a single, different subtypes or different cell types within the same lineage. So myeloid progenitor cells can differentiate into cells of a single lineage, or lymphoid progenitor also for that matter. So pluripotent would be for option 2. They can develop into any of the blood cells. So these are hematopoietic stem cells. Option B is the correct option in this case. Just give me a second. Getting calls. Okay, so Next question are for examples of secondary lymphoid organs. I did not get into this directly because this was already part of the lecture. So spleen and thymus, lymph nodes and thymus, spleen and gall, bone marrow and malt. So gall and malt are tissues. They are not exactly organs because they do not necessarily form a physical barrier, but they are considered as secondary lymphoid organs because they form a part of the immune system. So it is lymphoid tissue, so therefore it is included under the subtype of secondary lymphoid organs. So examples in this case would be spleen and gall. On the other hand, thymus, of course, thymus, bone marrow, these are primary lymphoid organs. So the correct option in this case are spleen and gall, gut associated lymphoid tissue. Then comes acquired immunity shows all of the following except, so we talked about the different features of acquired immunity as well. That A, it is non-specific to a particular antigen. It's diversity, non-self recognition or memory. So after all the discussion we have had, the correct option in this case would be non-specific to particular antigen. So it does show antigenic specificity. It is a crucial part of acquired immunity. So option A would be correct. Then comes question 6 on the right here. Choose the mismatch between these. So plasma cells, basophils, macrophages, and NK cells. So among them, you have to look at the matching that occurs between them. So among these, plasma cells are the activated or differentiated B cells which secrete antibody molecules. So that is the correct option. You have NK cells, which lies virus infected cells. That is true. You have macrophages and basophils as options here. You need to look up this option and see which of these are true. If you remember, I had discussed eosinophils, basophils, and neutrophils. Look up their individual functions. How are they responsible? I'm not bringing it up because this would be a very simple answer, but it is between B and C. And question 8 is for the term vaccination that was coined by which of these following. So vaccination, the concept of vaccination, as we had initially discussed, was brought about because of immunologic memory, which is a part of the immune system, which is a characteristic feature of the adaptive immune system. And that gave rise to the concept of vaccination, and it was coined by Edward Jenner. So you can go back into the history of it, that how did it discover, how was it at all understood that you can take the serum from one particular affected individual, inject that into another person, and then you see how that person becomes resistant or not resistant, but at least is sensitized to that disease, and they're able to tackle the same. So the concept of vaccination, it's been a while, but yeah, you can go back into the history of it. It's very interesting. And then you can see how that exactly came about. So with this, I'm coming to the end of my presentation. So now would be the time to go ahead and ask any questions if you have. So we have some time on hand. Please go ahead. And please let me know if you were not clear about any of the topics that I discussed today. Any questions, please raise your hand. Okay, if not, if anybody has any inputs that you want the classes to be, the discussion sessions to be carried out in a different manner, please let me know. You can maybe even drop an email or you can write it on the discussion forum. that you want more questions or you want more discussion. This was the first session, so I conducted it in this way. If you want more practice questions, maybe we can do that. I'll come up with more questions and then we can have an interactive session. But I would really urge everybody to go ahead and speak up. It will help in clearing up your concepts as well. And I'll get more material also if that is helpful. So if there are no more questions, then I'll end the meeting for today. You'll find the topics, the lecture uploaded on YouTube. You'll get the link for it, I think, sometime next week. And apart from that, the slides would also be available. And yeah, so for now, that's all I have. I'll get more questions for you guys. Please be more interactive. I would urge you to do that and go ahead and study from the textbooks if they're available with you. If not, please let me know. I'll upload the version of Kubi that I have. That's a very, very good reference book for your at least for your preliminary understanding. Okay, so if that is all, you can come up with questions next week. No worries. There's no hurry on it as well. But okay, thank you so much for joining. We'll again meet next Sunday at the same time. Please come up with questions or at least discussions. And thank you so much and have a nice day.
Thank you. Thank you. Thank you, ma'am.